All right. So I should please put on a camera. So actually in the previous class, we were discussing the kingdom Monera. One. Yes. So in the kingdom Monera, we discussed various things that basically when we talk of eubacteria, so we had two categories in kingdom Monera. We had the archaea bacteria and we had the eubacteria. So in archaea bacteria, we had three categories. Do you remember the examples of archaea bacteria?
What?
Aisha, audible? Yeah, am I audible to you, Aisha? I'm
Ma'am,
asking
you
you,
said that you will share the notes.
repeat again, please.
You said that you will share the notes.
Yeah, I told you that I'll share the notes, but actually the notes were not complete yet, so I did not share them. Today in the class, we'll complete the Kingdom Monera once, and then I'll be sharing the notes with you.
Okay.
Okay. So yeah, what were the categories of archaea bacteria? We studied three categories, three examples, not categories basically. The ones that, okay, do you remember something about archaea bacteria? What kind of cell wall were they having? Aisha, did you revise this lesson?
Okay, ma'am, I did not.
I do not hear you, Aisha. Are you speaking anything? Please put on put on your mic and speak.
Ma'am, you did not share the notes.
Yes, Aisha, I do remember I didn't share the notes, but I did teach you these things. So you were supposed to revise them from your textbook. Okay, no problem. If you haven't revised anything, we'll just continue ahead. We'll revise whatever we studied in previous class, okay? So basically, I taught you that the kingdom Monera has two categories, which is the archaea bacteria and the eubacteria. Yes. So when we talk about the kingdom Monera, so we studied that bacteria are the sole members of the kingdom Monera. That means in Monera, this kingdom, all the organisms are just bacteria. And bacteria are also the most abundant microorganisms. That means they are mostly, there are a lot of bacteria that are present in this on the earth. When we talk about habitat of Monera, so these live in extreme conditions. And why are they able to live in extreme conditions? Because they have a very complex structure of cell. Right? Then we studied three examples, which were halophiles, thermoacidophiles, and methanogens. So methanogens are basically the ones that are present in the cow guts and the marshy areas. Thermoacidophiles used to be present in the hot and the acidic environments, and halophiles were the ones that are present in extremely salty. So these are three examples of archaea bacteria. Then we studied about the cell wall, and the cell wall is basically composed of amino acids and polysaccharides. Then I give you two examples. These are the examples of polysaccharides. What were they? N-acetyl glucosamine and N-acetyl methacrylate. Then we studied that what is the cell wall? The cell wall is very complex in structure. So it basically helps the bacteria to survive in extreme conditions. 1 minute, Aisha, okay? Okay. So then we came to the complex structure of the cell wall of a bacteria. We studied that the cell wall structure is very complex in nature. Aisha, please put on your camera. Are you there in the class? Yes. We studied that the cell wall is very complex in nature, and its complex nature basically helps the bacteria to survive in extremes of habitats. Right? Then we studied shapes of bacteria. So, what were the four shapes of bacteria? Can you tell me the four shapes? I don't hear you, Aisha. Do you have some network issue? I'm unable to hear you.
Spherical, spiral, rod-shaped, and comet-shaped.
Yes. So what was the name of spherical bacteria? It was Coccus. Then for spiral shaped, it was Spirillum. For rod shaped, it is Bacillus. And for comma shaped, it is Vibrio. Then for spherical also, we studied two examples. It can be either Staphylococcus or Streptococcus. So what was Staphylococcus? If you remember, Staphylococcus was when the colonies of the bacteria are just scattered around like this. Right? Then it is an example of Staphylococcus. While Streptococcus, the colonies are arranged in a linear chain. All right. Then we have archaea bacteria. So archaea bacteria were basically one of the most ancient organisms. Like they were the first very primitive organisms to colonize the earth, and they had again a very complex structure, and they can live in variety of habitats. So we studied three examples, which were halophiles, thermoacidophile, and methanogens. Halophiles were the ones living in salty areas. Thermoacidophiles used to live in extremes of hot conditions and acidic conditions. Basically, hot springs is an example. Then methanogens are the ones that are living in extremes marshy conditions, and also they are present in the gut of ruminant animals. And what do they do in the gut of ruminant animals? What is the function of methanogens?
They are responsible for biogas.
For biogas synthesis. They basically form biogas from the animal tongue. Right? Then we have eubacteria. So talking about eubacteria, these are the true bacteria. Okay, eu means true. That means they're truly living structures. So what do they have? They have a motile. They have a motile body. So motile means the ability for locomotion is present. They can move from one place to another. And this is only present with the help of a structure known as flagellum. Okay. Then we came to an example of eubacteria, which is cyanobacteria. So cyanobacteria we'll study in detail now. So when you talk about cyanobacteria, the another name for cyanobacteria is blue-green algae. Okay, the cyanobacteria is also known as blue-green algae. And what happens? Another name is blue-green algae. And what happens? It has the ability to do photosynthesis. Okay, so that means cyanobacteria is photosynthetic autotrophic. Why? Because it has chlorophyll A molecule. The chlorophyll A molecule is present. So that is why it is able to do photosynthesis. Next, we came to its body structure. So cyanobacteria are basically unicellular organisms. And since they are unicellular, so what will they do? They will form colonies and they will live in those colonies. So they are colonial in nature, and they also have they form filamentous structures. So they are filamentous. Okay. And then we came to the habitat. The habitat is generally fresh water or marine water. So I gave you examples of fresh water also. Fresh water is basically like like lakes, ponds, and rivers. It's example of fresh water. Marine water, the example is seas and oceans. Okay, they can also be found sometimes in terrestrial habitats. Then we came to one important thing. Now, if you look at the diagram, now this is a nostoc. Now, nostoc is basically a cyanobacteria only. So look at the diagram. So these are the various cells of, this is one cell, another cell. So these are the cells, and all of they are forming a colony. So do you understand how they are colonious in nature? Because they are forming colonies. Now they have one very specialized structure which is known as heterocyst. What is the function of this heterocyst? Heterocyst is a cell that does the function of nitrogen fixation. Now what is the meaning of nitrogen fixation? Do you know? What is the meaning of nitrogen fixation? See what happens, the nitrogen is present in abundance in the atmosphere, but that nitrogen is not in the usable format. Okay, so what do the organisms have to do? The organisms have to basically the plants have to convert that atmospheric nitrogen into usable forms of like ammonia, right? Nitrates, nitrites, and ammonia. So what will they do? The process of converting atmospheric nitrogen into usable forms is known as nitrogen fixation. All right. So this is the function of specialized structures known as heterocysts. They do the function of nitrogen fixation. Now, this nostoc also has a gelatinous covering around it, which is known as mucilaginous sheath. Remember these words, very important. What does the mucilaginous sheath do? It is basically having the function of like protection. It is going to protect the entire colony and it will keep the colony intact. Like it is going to keep all the cells fixed at one place only. It will keep the colony intact and it will protect from desiccation. Right. So these are the two important structures of a nostoc. Then we have, so more examples of cyanobacteria can be like anabil. So two examples you should learn which are mentioned in your book, nostoc and anabil. Okay. Right, Asha? Do you understand this much?
Yes.
Okay. Now we come to the next topic, which is chemosynthetic autotrophic. See, we are studying about eubacteria. So first, according to mode of nutrition, if we classify, the first category is autotrophic. So in autotrophic, you can have two modes, either photosynthetic autotrophic or chemosynthetic autotrophic. So what is the example of photosynthetic autotrophic? We studied just now. What is the example?
Worms,
Cyanobacteria. What happened, Aisha? Are you able to understand or not? If you look carefully, what we have in eubacteria, the mode of nutrition can be diverse. It can be autotrophic or heterotrophic. If the mode of nutrition is autotrophic, whether it's using photosynthesis or it's using chemosynthesis as its option. Aisha, do you hear me?
yes,
Okay, so I'm talking about cyanobacteria. So cyanobacteria are basically photosynthetic autotrophs. Now we come to chemosynthetic autotrophs. So what does chemosynthetic autotrophs do? They will basically, okay, see, what is the main function of the autotrophs? To produce food, to make food. So what are they doing? The chemosynthetic autotroph will basically, they will oxidize the inorganic substances. Like they're using the nitrates, nitrites, and ammonia. They will use these substances for ATP production. All right. The main thing is ATP, right? They have to produce ATP. So what are they doing? They're using these nitrates, nitrites, and ammonia. These are the three important things that you should know. They're using these substances in ATP production, and the chemosynthetic autotrophs also has a role in recycling nutrients. Like they recycle nitrogen, phosphorus, iron, and sulfur. So this is one important point. Then when we talk of heterotrophic bacteria, these are one of the most abundant organisms, right? They are present everywhere on the earth. So these are decomposers. Obviously, heterotrophic nutrition, decomposition is an example of heterotrophic nutrition. So these are decomposers. And what do they do? Three important functions is that we can use these heterotrophic bacteria for the production of curd. Have you heard of the name lactobacillus?
yes,
Yeah, so lactobacillus, what does it do? It is a heterotrophic bacteria and it will produce milk curd from the milk, right? Then we have antibiotics production. Like these heterotrophic bacteria can also be used to produce antibiotics. Then we have like nitrogen fixation to increase soil fertility. Again, what is the meaning of nitrogen fixation now? Can you tell me?
atmospheric method.
Speak again, Aisha. Please put in a mic and speak. Don't put it off. What is nitrogen fixation?
Atmospheric nitrogen is like usable.
Conversion of
Atmospheric
nitrogen into
nit-
usable form. Right? So
Yes,
that is known as nitrogen
yes,
fixation. So they will do nitrogen fixation and it will increase the soil. See, obviously, if the soil contains more amount of usable nitrogen, then it means it is fertile. So that is why they're also increasing soil fertility. So these three functions, will you remember? Then, okay, many heterotrophic bacteria can also be pathogens. Okay, like we have some diseases like we have cholera, typhoid, and citrus canker. These are some of the diseases that are caused by bacteria. Okay, cholera is cholera, typhoid, and citrus canker. So remember these three things. So these four are the important points about heterotrophic bacteria: curd production from the milk, antibiotics, and nitrogen fixation, and finally as pathogens. So these 4 points you should remember. Then we finally come to reproduction in bacteria. How does a bacteria reproduces? So there can be three modes of reproduction: fission, spore formation, or sexual reproduction. Now, what is the meaning of fission? See, let's suppose I have a bacteria over here, and what does this bacteria do? It will divide itself into two daughter cells. So I have one daughter cell over here, one daughter cell over here. So from one bacteria, I have two bacteria. Now this will again divide itself into two, and I have four bacteria now. Right? So from one bacteria, I have produced four bacteria. So this is fission. Now what is spore formation? Spore formation is, let's suppose I have a bacteria over here. What it will do, it will form a spore of its body. It forms a spore. And now when the conditions are favorable, the spore shall germinate and form various bacteria. Then sexual reproduction, the example is conjugation. So what happens in sexual reproduction? Like this is a one bacteria. This is another bacteria. They will join together and they will transfer like the DNA from this is the DNA of this bacteria, this is the DNA of this bacteria. And they will just transfer the DNA from one bacteria to another. So this transfer of DNA is known as conjugation. Okay, Aisha, you understand this? Do you understand this? Then we have a case study of mycoplasma. Now, what are mycoplasmas? Mycoplasmas also belonging to the kingdom Monera also. And they are the smallest living cells. They are very small. They are microscopic. And they do not have a cell wall around them. Right? Also, the mycoplasmas will survive without oxygen and they are pathogenic in plants and animals. So, what all conditions do we have over here? They are smallest known cells. They do not have a cell wall. They can survive without oxygen and they are pathogenic. You understand all these 4 points? So you can do one thing, Aisha. Please go through them one by one once again. And you can copy the notes right here only so that it helps you to understand and know what all topics you are having a doubt in. Okay. So please start copying.
then
Okay. So you come to this part now. Do you have your NCRT textbook with you?
no,
Do you have your NCRT textbook with you?
no,
You don't have it. You don't have it. You've not purchased it.
No, I have heard this.
So where is it, Aisha?
But no, I don't have.
Okay, no problem. Whenever you have your textbook with you, please sit with your textbook, okay, from the next class onwards. So you can write down the functions of these also, like what is the function of heterocyst and mucilaginous sheath. In your NCERT textbook only, make these arrows and just write them down. You have a lot of space over there. This you can leave for now because I have already written this in notes. Now you can come to this part, chemosynthetic autotrophs.
Then
Yes, Tanisha. Can you please copy this last part also? Then we'll move to the next thing.
then
Completed? Shall we move ahead? Okay. So now we come to the next kingdom. So till now we have studied one kingdom, which is the kingdom Monera. Now we come to the next kingdom, kingdom number 2, which is Protista. Now what happens? What are protistans? Now if you remember that table, so in that table it was written that these are eukaryotes. Monerans were prokaryotes and protistans and all other kingdoms are eukaryotes. So protistans are basically eukaryotes, but they are single-celled organisms. Generally, if you look at all the other kingdoms, they are eukaryotes, but they are multicellular. But when you talk of protista are eukaryotes and they are single-celled organisms. Right. Also, the protistans are primarily aquatic. That means what is their habitat? They are mainly living in water. They can be living in fresh water, they can be living in marine water, but they are primarily aquatic. They're not found on land. See, Aisha, whenever you hear the word aquatic as the habitat, you try to associate them to be a primitive type of organism. Okay, I'm not talking about fishes. See, fishes are complex organisms. They are not primitive. They although they live in water, but mainly the primitive organisms have aquatic habitat only. Monarchs are primitive, so mainly they live in water. Similarly, protistans are primitive, mainly they live in water. Okay. So what kind of nucleus do they have since they are eukaryotes? They're going to have well-defined nucleus. And they're also going to have various other organelles which are to be membrane bound. Right? Now, again, they have a cilia as well as flagellum might be present. It might not be present also sometimes, but most of the time it's present only for locomotion. Reproduction can be of both types. It can be sexual also, asexual. Okay. Now what we have, protistans have various categories. It is 5 categories. So one by one, we'll just study those categories. So when you talk about chrysophytes, the first category. So chrysophytes are basically known as the chief producers in ocean. Why are they known as chief producers in ocean? Because chrysophytes are able to do photosynthesis. Okay, and since they are present in very large numbers, so the amount of photosynthesis and the food produced is also very large. Hence, they are known as chief producers. Now, the chrysophytes are basically examples are diatoms and golden algae. Golden algae are also known as desmids. So remember these two examples. You have diatoms and golden algae. Then what is the habitat? It can either be fresh water, it can be marine water. Now, chrysophytes will float passively in the water current. What is the meaning of floating passively? That means they are not using any energy of their own. Like wherever the water current is taking them, they will just flow with the water current. So that is known as passive floating, right? Active floating is like when like fishes swim. Fishes will swim in a water in whatever direction they want, right? But when you talk about chrysophytes, they do not have any direction of their own. Wherever the water will flow, they will float with them. So that is known as they were floating passively in the water currents. Then what is the size of chrysophytes? They are very small, tiny structures. Basically, they are microscopic only. Are they able to do photosynthesis or not? Yes, they are photosynthetic. We studied protistans are photosynthetic, right? So this is an example of protist only, chrysophytes. Then how is the cell wall? Now, cell wall is very interesting for chrysophytes, and also you need to learn this thing very well. That the cell wall, okay, have you ever seen a soap box? A soap box with the lid that closes something like this, like it closes like this, a soap box. Have you seen soap boxes like this?
Yes.
Okay. So what happens? Its cell wall is also in a structure of a soap box only. Okay, it has like two shells, and inside those shells, the chrysophyte's body is present, and its cell wall is basically like two thin overlapping shells. And the cell wall is basically made up of silica. Now, this thing is very important. See what happens, there's a property of silica, that silica is indestructible. That means silica cannot be destructed. Right. Now what happens, since silica cannot be destructed, now over the period of long time, the chrysophytes kept on dying, and the cell walls kept on accumulating in the water body. On the surface of the water, their cell walls just kept on accumulating for a very long time. And after approximately like billions of years, what happened? Those silicon cell wall deposits formed diatomaceous earth. Now, what is this diatomaceous earth? Basically, the surface of the ocean that contains a lot of amount of silicon cell walls of the diatoms. Now, this diatomaceous earth is basically gritty in nature. Okay, and what does this help? How does this gritty nature of the diatomaceous earth help us? It can be used in polishing and filtration of oils and syrups. So when we are filtering oils and syrups to be part of this diatomaceous earth, they will be filtered very. 1 minute, Aisha. So this diatomaceous earth can be used for various purposes like polishing, filtration of oils and syrups, etc. So an example of chrysophytes is navicular. Okay, you need to remember these examples. Then we come to dinoflagellates. Now, what are dinoflagellates? This is an example. This is a figure of a dinoflagellate. Now, dinoflagellates are again going to be marine only, and they will be photosynthetic. That means they will form their own food. The color of the dinoflagellate is like diverse. It can be of various colors: yellow, green, brown, red, and blue. Now, why does this color arises in the dinoflagellates? Basically, they have various different types of pigments in their body, and the type of the pigment will define the color. It will define the color of the body of the dinoflagellate. So it can be of these various five colors: yellow, green, brown, red, and blue. Okay. Now, how is the cell wall of the dinoflagellate? The cell wall is basically cellulose. It is made up of cellulose and it is very stiff. That means very hard. Okay. Now the flagella. Now this is very important. The dinoflagellates have two flagella. Hence the name dinoflagellate. Dino means means two, and flagella means having flagella. So dinoflagellate will have two flagella. What kinds of flagella? See, one of the flagella is like this. This is a longitudinal flagella, and the other flagella is like this. This is a transverse flagella. So it has two flagella. Very important thing. One is longitudinal while the other is transverse. Now, an example of this dinoflagellate is a red dinoflagellate, which is known as coniolax. Now, what does this coniolax do? It is red in color. The coniolax is red in color, and sometimes this coniolax may rapidly multiply to form many, like many colonies of coniolax. Right. And finally, what happens? That rapid multiplication will make the water body to appear red, and that is known as red tide. Okay. So red tide is
Okay.
associated with the red dinoflagellate. Now, also these conjuncts might produce a toxin which is harmful for fishes, right? And the fishes might also die. So during a red tide, fishes might die. Why? Because that red tide contains goniolax, which is producing toxin, okay, which is harmful for the fishes. So do you understand these two things?
Yes.
Okay, so please quickly copy these points about protista. Aisha, you copy?
Yes.
Okay.
Then
Yes, Aisha copied. So come to chrysophytes now.
Then
Yes, Asha, done. Yes, Aisha, have you copied? Aisha, please tell me, have you copied? Aisha, are you there in the class? Aisha, do you hear me? Aisha, I guess you have some network connection issue.